I'm going to go over the specific defense, and so we call this the immune system. And so we're going to start with the organs. So when we say the immune system, we're talking specifically about the specific defense, and this is going to fight off pathogens, and it's also going to help prevent the growth and spread of cancer. There's many organs that are part of the immune system. We have bone marrow, the thymus, lymph nodes, tonsils, adenoids, and spleen. And I'll go through and explain the role that each of those organs plays. The white blood cells that take part in the immune system are called lymphocytes. So starting with the bone marrow and the thymus, the role of the bone marrow is to make new white blood cells. Now, there's two main types of white blood cells that participate in specific immune responses, and one of them is going to stay in the bone marrow to mature and to specialize. The other one is going to go to the thymus to mature, and they're going to be named based on where they mature. There's B cells and T cells. I'll talk about those later. Okay, then we have lymph nodes. You have lymph nodes all over your body. In green, which you can see here, is your entire lymphatic system. If you remember the lymphatic system from the cardiovascular chapter, and then the little tiny dots that you see, they're concentrated in your neck up here, in the armpits, and in the groin area here. And lymph nodes contain large amounts of white blood cells, and this is going to filter any pathogens from lymph, which is that fluid, as it gets returned back to the bloodstream. Next, we have the spleen. So if you want to know where the spleen is, here's your liver, which is located right underneath your diaphragm. And then the spleen is on the left side of your body, so it's on the right side of the picture, under the rib cage. And a lot of times in human anatomy, we talk about how different organs are about the size of your fist. And that's usually a good rule of thumb because your hand size is proportional to your body size. So the bigger you are, the bigger your hands are, and the smaller you are, the smaller your hands are. So we use that to describe the size of the spleen, your kidneys, the uterus, and the heart. The spleen is the largest lymphatic organ, and its role is to store healthy blood cells and break down old blood cells. It also helps to develop lymphocytes and other white blood cells. You don't, however, need your spleen to survive. There's sometimes when you have a crushing accident, like maybe a car accident or something like that, and you can have a large amount of damage on the left side of your body, and it can sometimes crush the spleen, and they do what's called a splenectomy. And in surgical terms, if you have ectomy at the end of something, it means you're cutting it out. So you don't have to, you don't need your spleen to survive. The spleen can also collect pathogens from the blood, and the lymphocytes that are in the spleen can attack those pathogens. Your adenoids and your tonsils, you can see here, are at the back of your mouth. They're lymph tissues that are in your nose and your throat. You can see the tonsils here on either side. These look like they're inflamed because they're not supposed to be that large. And so if that is the case, sometimes what they'll do is a tonsillectomy, which they'll cut these out as well. Okay, so for lymphocytes, like I mentioned a minute ago, you have two main types of lymphocytes that participate in specific immune responses. You have the B cells. Of course, they're made in the bone marrow because all red and white blood cells are made in the bone marrow, but they're called B cells because they complete their development in the bone marrow. The second type of lymphocyte is called a T cell, because even though they're made in the bone marrow, they go to the thymus to complete their development, and so they're called T cells. Now, the reason why we distinguish between these two types of cells is not just because they mature in different places, but it's because these two cells are going to have completely different approaches to dealing with a pathogen or an infection. And so we have to talk about them differently. They have completely different responses. So the reason why, sorry, this is like the sound is in my ear, but I can't turn it down. Never mind. So what you're looking at here, this is how your body identifies pathogens by the antigens on the surface. So in this, the green blob is the pathogen. If you imagine it's like the coronavirus, right? And so you've seen pictures of the coronavirus and it has like these little, looks like a ball on a stick on the top. That's the antigen. The protein spikes is what they talk about. And so you see how, remember I always tell you that form or the structure means everything in biology. It always has to do with a physical fit, like a lock and a key. So you see here how your white blood cell has the correct shape of receptor to match exactly with this particular pathogen. That's what we mean when we say specific defense. It has to physically match the receptors on the pathogen in order for your body to understand, okay, this is the shape that we need to match. And then you launch your B and T cell response according to that specific shape. So when we say things like the coronavirus is a novel virus, novel means new. It's never before been seen in the human species. And so it's going to have these antigens. You can see them up here, the antigens. We talked about that in the last chapter when I mentioned blood type. Antigens, remember, stand for antibody generating substance. They are could be spike proteins, could be carbohydrates, things that basically your body doesn't recognize as belonging to you. So then it can launch an attack against it. So lymphocytes provide a specific defense because they recognize foreign invaders. So like I said, coronavirus is a novel virus because it's never been seen in humans. And this is why, especially being as contagious as it is, it becomes a worldwide pandemic because as it's spreading throughout the world, our bodies, basically, if we get infected with it, our bodies are trying to figure out what shape that spike protein is, because then it can launch the correct specific response. And that's what it means by specific immunity. So like I said, an antigen is anything that that your immune system doesn't recognize as being part of you. So antigens are going to cause lymphocytes to react. A lot of things can be antigens. It's not just virus. You also have parts of pathogens, which is one of the ways that we develop vaccines is that we can take, say like a flu virus, even like H1N1, the bird flu, that was like 2012, I think, something like that. And we can take a virus and we can, it was called heat kill. I think I talked about that last year when I was talking about the Hershey Chase experiment. Griffith's experiment when he heat killed like the S bacteria and they changed into R bacteria. At high temperatures, remember, DNA denatures, it falls apart. And so the only reason why viruses are dangerous is because of their DNA or RNA. You have different kinds of viruses. There's DNA viruses and RNA viruses. But if you expose the virus to heat and you cause the nucleic acid to denature, then you can inject the deactivated virus into the human body because your immune system doesn't respond to the DNA or the RNA. It's responding to those surface proteins. Bacterial toxins can also be antigens, insect venom, or even pollen. And that's when you start getting into allergies and like inappropriate immune responses. Anything that's not a natural part of your body can become a pathogen. So this includes transplanted tissues or organs, or even transfusing blood that's the wrong type. And this is why I said before, if you know your blood type, knowing whether your Rh factor is positive or negative is just as important as knowing whether you're AB, or O. So it's important to know, like, not only are you type O, but are you O positive or O negative? Because that third antigen is just as important as the A or the O. The transplanted tissues and organs, this is why doctors have to do thorough testing beforehand. They have to match tissue types, which is a completely different scenario than just having to match blood type. Just because you're the same blood type doesn't mean that you can donate an organ to somebody that has the same blood type. You have to match tissue type. So it's a more specific kind of testing. And it's also the reason why people are on the wait list for organ transplant for years, waiting for an organ that matches their specific tissue type. Even then, after transplantation occurs, the patient then has to be put on immunosuppressants, meaning that they are put on medicine that suppresses or slows down their immune system in order for that organ to be accepted. And even then, sometimes the patient experiences organ rejection. Is it like it's, even though they're tissue matched, the body still recognizes it as like, this doesn't belong to me, it doesn't belong here, and it starts attacking the organ. If that's the case, then they have to be put back on the transplant list and try to find a better match. It's a complicated process. So when lymphocytes recognize an antigen, they're going to bind to the antigen, like you saw in that video where the pathogen with the pointy antigen matched with the surface receptor. And that is going to start the immune response. And there's going to be, like I said, there's going to be two sides to the immune response. They're both going to happen at the same time. Obviously, I can't explain both at the same time, so I'll explain them one by one. Your lymphocytes that are made in response to a pathogen, they're all going to have receptors of the same shape because this is a specific immune response. It is made specifically for this particular pathogen. It's not going to work against, like a coronavirus vaccine is not going to work against the flu because a flu has completely different spike proteins than coronavirus. This is also the problem that we come across, especially right now with the Delta variant becoming the most dominant variant in Thailand and pretty much all over the world, because I was reading an article last week that said that the Delta variant has 7 mutations in the spike protein, which is why you're seeing people that are fully vaccinated, even people in the US that were vaccinated with Pfizer with both shots, still turning up positive. So you can still contract or you can still get infected with the virus. And it's important to remember that even if you're vaccinated, even with the best vaccines, they're not 100% protection against being infected. The only thing vaccines do, which is part of what I'll talk about on Friday, but what a vaccine does is it kind of primes your immune system and gets it ready in case you do come across this particular virus that the vaccine is made for. So your body has millions of different kinds of lymphocytes. All of the diseases, I guess disease is kind of a strong word, but all of the illnesses that you have experienced over your entire lifetime so far, you more than likely have lymphocytes left over from that illness. So think about how many types of cold, or how many times you've had a cold, how many times you've had the flu. Every single time it has been a different version of that virus, which is why you get sick again. So if you think about, like, when the flu is going around at school, like when we are actually in person at school, and one person starts getting sick, right? And then usually they will pass it on to at least a few people in the class because you guys are in the classroom all day together. But it doesn't come back around and reinfect like patient zero. It's like if I get sick and I pass it to you guys, I don't get reinfected and get sick again because my body's already fought it off. Even though I'm constantly exposed to it every day, my immune system has already built a response to it. So it kills the virus off that you guys are shedding back to me before I get sick again. And so that's basically what vaccines are there for, is to kind of like, it's like a cheat sheet for your immune system. It's like, here's what we're fighting. And when you see this in real life, like, go attack it before it makes you sick. So you might read stories. I don't know how much of you guys like read the news, and everything's about COVID these days, but even if you read stories, like there was a story in the Bangkok Post the week before last, I think, that they said that 618, I think, so around 600 medical workers tested positive for COVID, even after they had both shots of Sinovac. It doesn't necessarily mean that they're all going to get sick and die from it. So the vaccine, one, isn't 100% protection against being infected. The purpose of a vaccine is to try to prevent a serious illness as a result of being infected. So I know that the news focused on one particular nurse. I think she was like 28 years old, and I think she died from it. And that, that happens because we all have different immune systems and we all have different kind of natural protections. Even with vaccines, nothing is, like I said, nothing's 100%. But being vaccinated means that you are more protected against developing a serious illness and having to be hospitalized because of it. So that's just different things to keep in mind as we learn about these things. Okay, so the specificity of your immune system is caused by the specificity of these antigen receptors. This is another reason why, let's take, going back pre-COVID times, if you ever got your flu shot, if you look at the brochure that they, I don't know if they give it to you or if it's there just for you to look at it, they'll actually list the strains of flu virus that are included in that vaccine. And it's usually like 4 or 5 strains of the flu virus that were circulating the previous year. So the hope there is that if you get some kind of related flu that has maybe similar shape, that you can have enough of a response to protect against it. But again, viruses mutate all the time. We've already seen, obviously, with COVID being the number one thing in the world at the moment, we've already seen was that alpha, beta, I don't know if they had a gamma, but now you've got Delta, and Lambda is a new one as well that I've heard is around South America, I think. So these are all mutations, which is totally normal for viruses, which is why it's so important for all of us to try to get vaccinated as soon as possible, because the longer this virus circulates in the human population, the more likely it is that you get more and more variants. Okay, so what is the immune response? Like I said, 2 approaches. You have the cell-mediated immune response, which is the T cell half of it, and you have the humoral immune response, which is the B cell side of it. And like I said, there's 2 completely different approaches, and but they happen both at the same time. Both of these immune responses are controlled by a cell called the helper T cell. So there's going to be a series of events that occur first, and then you get to a point where the last step will kick off these B cell and T cell responses. So the first thing that happens is a macrophage. Remember, I talked about macrophages. These are like the Pac-Mans of the immune system. They are circulating through your blood, and if they encounter a pathogen, the macrophage is going to engulf and destroy it. But once it's destroyed, it's actually going to display pieces of the antigens on the surface of its own membrane. Remember when I said the natural killer T cells, how do they recognize which cells are infected and which ones aren't? The cells that are infected display the antigens that match the pathogen. And this is what I mean by specificity. So macrophages are both part of your non-specific defense, but they are also the first ones in the step for your specific immune response. So macrophage engulfs a pathogen, and the macrophage is going to have these specific antigens on the surface of its own membrane. Now, helper T cells. The helper T cells are also kind of floating around in circulation. And when a helper T cell that has the matching receptors meet the macrophage is going to release a cytokine, which is a chemical. And the name of the chemical that the macrophage releases is called interleukin-1. You can also call it IL-1, is fine as well. And so that is a signal to the helper T cell that this is the correct shape. This is the shape that we need to fight. And in return, the helper T cell releases IL-2. And IL-2 is the one that's going to stimulate the B cells and the T cells. Okay, so all of these things are the ones that happen first. Macrophage encounters a pathogen, so it comes across the coronavirus, it engulfs and destroys it, but it's going to have the spike proteins on the surface. Your helper T cells, if you don't have any helper T cells that have matching receptors, this is when your immune system starts pumping out helper T cells with different receptors on it, and then that's the period during which you develop symptoms and you become sick. That time that your body is trying to figure out, what is this shape? What is this antigen? I've never seen this before. How do we fight it? So that's the period of time where you develop symptoms, get sick. Finally, when your body makes a helper T cell with a receptor that matches, that's when the macrophage releases interleukin 1 to be like, yep, that's the one. And then the helper T cell releases interleukin 2. Once that happens, then we get into our B and T cell response. So I'm going to start with cell-mediated immune response, which is the T cells. So the last thing we talked about was that interleukin 2 is released by the helper T cells. So the interleukin 2 makes two different kinds of T cells divide rapidly. We have cytotoxic T cells and we have suppressor T cells. Now, if you just look at the word cytotoxic and you break that down, I'm sure you all understand what toxic means. And remember that cyto means cell. So cytotoxic T cells are also the natural killer T cells, but these are lymphocytes. They're not the same natural killer cells that run around and actually, they might be the same. I think they might be the same. But the cell-mediated immune response, or the T cell response, is not going to go after the pathogen directly. It goes after your cells that are infected. The response that goes after the pathogen directly is the B cell response, the humoral. Because if you think about when you get infected with a pathogen, it's not enough just to go after the pathogen itself, because if you have infected cells, specifically for viruses, because viruses hijack your cells and then your cells become little virus factories. So if you only take care of the pathogen, then you're still going to have your little virus factories that are making more and more virus. At the same time, it doesn't make sense just to kill the infected cells because you still have the pathogen running around in the body. So you need to do both. You need to deal with the cells that are infected, and you need to deal with the pathogen directly. The cell-mediated immune response deals with infected cells. So here is a picture. You can see the cancer cell, and the cytotoxic T cell is attacking the cancer cell. This is what I mentioned last week, that your immune system can keep cancer cells in check to a point, depending on how quickly they're dividing and whether or not it spreads. As I mentioned last week, because your infected body cells have the antigen on the membrane, the cytotoxic T cells attack and puncture the membrane. Any sound? No sound. Okay. All right, so then I will narrate, since there's no soundtrack to this video. So you've got the healthy cell and the cytotoxic T cell. You can see the MHC class 1, that is what they call a self-identifier. That's how the cytotoxic T cell can recognize. And now you see this virus-infected cell and how the surface protein is going to signal to the cytotoxic T cell that this cell is in fact infected because it has the viral antigen on the surface. So there are your cytokines, your interleukin 1 and 2. And now you've got a protein, perforin, that's going to make the cell membrane of the infected cell more permeable. You want to make it more permeable because the cytotoxic T cell is going to then release the enzymes. Either the perforin pore is going to create more pores in the infected cell, and then you've got granzyme. It's an enzyme that's going to destroy the infected cell. And then the cytotoxic T cell can move on to the next infected cell. So hopefully when I said last week that the cytotoxic T cells or natural killer cells puncture the membrane, you didn't think that it was like they run around with like a little sword and like physically stab the membrane. They do it through enzyme action. Right, so that's the cytotoxic T cell part. Now the suppressor T cells are going to be there to shut down this half of the immune response after the pathogen is cleared. So cytotoxic T cells are like crazy soldiers that just run around and like kill all the infected cells, or like brainless, or like you're infected, die, you're infected, die, you're infected, die. And then finally, when the pathogen is cleared and there's no risk of having more cells being infected with pathogen, then the suppressor T cells are like the diplomats that come and like, okay, let's talk and let's go back to like being all calm. So that's the cell-mediated immune response. The second half of the two-prong attack is the humoral immune response, which deals with B cells. So again, we go back to the beginning where you've got the macrophage. Macrophage engulfs the pathogen, displays the antigens, helper T cell that matches, binds with the antigen. The macrophage releases interleukin-1, which causes the helper T cell to release interleukin-2. Now, in this case, interleukin-2, besides stimulating the cytotoxic and suppressor T cells to divide quickly, it's also going to stimulate the B cells to divide quickly, but it's also going to make the B cells differentiate and become what's called a plasma cell. A plasma cell is highly specialized, and they make defensive proteins and secrete them into the blood. These proteins are identical to the plasma cell's antigen receptors, so they match the antigen on the pathogen, and these proteins are called antibodies. And if you've read anything about coronavirus and how we're testing and things like that, you can do a blood serum test where they test for antibodies. It's also, if you read about like Sinovac, there was an article in the Bangkok Post, I think last week as well, that talked about if you get vaccinated with Sinovac, that every 40 days, the number of antibodies in the blood decreases by 50%. So every 40 days, your antibodies are halved, which to me does not sound good. Antibodies are Y-shaped molecules, and the two arms of these Y are identical. They attach to the same antigen. So you imagine one antibody particle can attach to two different viruses because you've got one arm, you got two arms, both arms match the antigen, so it can attach to two viruses. And one plasma cell can make up to 30,000 antibodies a second. So when you get sick, you can imagine how many millions, like hundreds of millions of antibodies that are running around attaching to these viruses. So how does that help? Because antibodies don't destroy the pathogens directly, but the way that the virus infects your cell, as you saw in that very first video, it has to attach to surface receptors on your cell. And it does that through the antigens. If the antigens are all being taken up with antibodies, there's no way that the virus can attach to your cell. And if there's no way it can attach, then you can't get infected, like your cell can't get infected. It also helps by creating, what do you call it? I'm like losing the word here. It creates clumps. Remember I said that the Y shape, each arm of the Y shape can attach to a different virus. So imagine if you've got millions of antibodies attaching to all of these viruses, it creates these clumps. And that triggers your non-specific defense because you've got your non-specific white blood cells, like your neutrophils that are running around. And if it sees kind of like groups of things going on in the blood, it's like, nah, that's not right, because blood is supposed to be nice and fluid. Right, so prevents it from entering the cell, but it also, clumping, I guess is the word I was looking for, causes clumping of pathogens, and that triggers the macrophages and your neutrophils to engulf them all. So here we've got just the blood flow with the red and white blood cells. And there That's the Y shape. So you can see it attaching to the antigens. Okay, the Y shape's your antibodies. So these are the pathogens, and if the pathogens are completely covered by antibodies, that's going to trigger your macrophages to come and engulf them. But this is the way of dealing with the pathogen directly. So the T cell response deals with your infected cells, and the B cell response deals with the pathogen directly. Now, the last thing for today is talking about primary and secondary response. After your infection is cleared, all of the infected cells are killed, the antibodies and the non-specific white blood cells have cleaned up all of the pathogens from your body. The immune system response shuts down. Most of these white blood cells that were created are going to die. But some B and C cells, some B cells and T cells stay. Oftentimes they're there for life, and these are called memory cells. Memory cells are your body's long-term protection against reinfection by a specific pathogen. So because of memory cells, you usually don't get most infections twice. So like I said, all of the times that you have been sick, it has most likely been from a different version. So for example, the common cold. The cold is caused by a group of viruses called adenoviruses. Adenovirus. And this is just a group of viruses that are responsible for causing the cold. Now, for the viral vector vaccines that we're using for coronavirus, that is AstraZeneca and Sinovac, these are taking the adenovirus and genetically modifying it to look like the coronavirus. So remember when I said that the DNA is really what's important. So if you have your virus, this is your cold virus, and the cold virus has your DNA. So this is DNA to cause a cold, but they make it look like corona. Because as you just learned today, your body doesn't respond to the DNA, it responds to the proteins on the surface. So you're taking a relatively harmless virus and you're making it look like the big bad coronavirus. And so that way your body launches your cell-mediated and your humoral immune responses. So you create the helper T cells to match this specific shape. And when it's finished, if you do get infected with the real coronavirus, then your body recognizes it right away because it's able to identify what it is. If you've never had coronavirus before and you're not vaccinated, your body spends all that time trying to figure out what that is. Once it's identified, then your body can try to fight it. Now, depending on people's immune system and their overall state of health, determines how seriousness of an illness they're going to experience. Obviously, coronavirus infects the lung cells first, which is why people have problems breathing, and they might have to be ventilated on the machine and all of that. But once your body figures out how to fight it, then depending on how much damage the virus did already, then you tend to get better. So if you're exposed for the second time, your memory cells recognize the antigen immediately, and your immune response is like more than twice as quick, and it kills the pathogen before it can cause the disease. So we have the primary immune response, which is the first time you got infected, which is like you're going to feel some kind of symptom. The severity of the symptoms, like I said, is completely individual and based on your overall health and genetics and all that kind of stuff. Every reinfection after the primary response is called your secondary immune response. It is more powerful and it is faster. So your body is going to produce tons of B cells and T cells. The B cells are going to start pumping out the antibodies, and your cytotoxic T cells are going to run around and kill any cells that are infected the second time.